Hi, everybody. In this review topic, we're going to jump off of what we left off with almost in the last section, where we were looking at the structure of the membrane. This finishes off Unit 2, where we're looking now at the function of the membrane. So how does that structure of a phospholipid bilayer with proteins embedded in it lead to its selective permeability? And that's what we're going to look at in this topic. So some things to remember. We always look at active transport versus passive transport. Passive transport depends on concentration gradient. So things are going to move from a high concentration to a low concentration. In active transport, energy is going to be used, specifically ATP is going to be used in order to move those substances from a low concentration to a high concentration. Now, some questions to maybe get you thinking about what you could possibly see on the AP exam. If a molecule is in a higher concentration outside the cell than inside the cell, which direction would it move? And it might be helpful to draw a little sketch of this. So looking at just dots on the outside of the cell compared to the inside of the cell, we would see that there are more outside, and by passive transport, they would move into the cell. Number 2, we're going to review here in a second on the next slide. So what type of molecules can move through the cell membrane versus what has to move through the proteins, etc. We'll also review the terms hypertonic versus hypotonic, and then we'll use that calculation of water potential to discuss that in this video. Something that you might also see is number 4, where they're saying, okay, if there's a situation where there's a change or the condition is different or there's a new environment, what's the cell going to do? So for example, if a cell is normally living in an isotonic solution and then that solution changes to hypertonic, what's going to happen to the cell and how is the cell going to try to respond? 5, now remember, we don't have to graph anything, but I have seen tons of questions in the past where they give you data from dialysis tubing experiments, or if you remember the potato experiment where you soak them in different colored water. I've seen tons of questions on the AP exam about that. We are going to look at one, but again, you won't have to graph this. You may just have to analyze it on this year's exam. As I said, number 6, we'll also look at reviewing that formula that's on your formula sheet to look at water potential. Okay, so again, as you saw in the last 2 topics, there's a list of important terms for you to review. Again, take some time, review these, and then at the end of the video, if there are some that we didn't hit or there are some that you're still confused on, reach out or attend the Zoom on Thursday and we can go over those as well. So first, let's take a look at how the structure of the membrane relates to the function and its selective permeability, and how the charges of molecules, the polarity of those molecules, determine which way those molecules are going to enter or exit the cell. So in the last video, we reviewed that the phospholipid heads are polar, so they are hydrophilic, and we have those 2 layers of hydrophilic heads. The interior of the membrane has the hydrophobic or the nonpolar tails. And then these proteins kind of provide a safe hydrophilic pathway for molecules to pass through. So let's review our different types of substances that could potentially be passing through the membrane. This chart up above looks at ions, large food molecules, liquids, and gases. And all of these things are substances that the cell needs to either have enter itself for use or for these things to leave itself because they're waste products. So looking at the ions, we know that ions are small because they are just one atom, but they have charges on them. As you know, things that are charged or polar cannot pass through the phospholipid bilayer itself. Because the interior of the membrane is nonpolar and non-charged, these substances are repelled by that interior part. So small ions cannot pass through the phospholipid bilayer. They, if they're passing through passive transport, can move through gated ion channels. So they can move through just through passive transport. Or if you think back to when we talked about the sodium-potassium pump, that's a form of active transport that uses ATP to establish a concentration gradient of sodium on the outside of the cell and potassium on the inside of the cell. So ions, either A, if they're moving passively, are going to move through the integral protein channels, or B, if they're moving through active transport, they're going to be moved through those membrane pumps, right? The sodium potassium pump, the hydrogen ion pump, etc. There's chlorine pumps. So those are the two different ways that ions move into or out of the cell. Food molecules, think of glucose or big carbohydrates or big proteins or big phospholipids or other types of lipids. They are too large, first of all, to pass through the phospholipid bilayer. They just can't fit. Most of these, like glucose and sugars and proteins, are usually polar. So they have two things going against them. They're charged and they're large, so they can't fit through the phospholipids. These can enter or exit the cell through either, if they're small enough, they can enter through the protein channels, or if they're too big, they can enter by endocytosis, right? When the cell kind of engulfs those molecules. The opposite would be true when these molecules are leaving the cell. If they're leaving the cell, they can exit either through those protein channels or they can be released from the cell through exocytosis. Our next topic are liquids, such as water. That's osmosis, right? Water is small, but it is polar, so it cannot fit through the phospholipids. It must enter and exit the cell through special proteins called aquaporins. So they're just a specialized channel protein that's made just for water. And we'll talk about osmosis coming up later on in the video. Gases like oxygen and carbon dioxide, as far as we talked, these are the only things that can pass through the membrane through the phospholipids because they have two things going for them. They are small, so they can fit, and they are non-polar, so they can shimmy through the phospholipids. They can go through without being repelled. So all of those things, we kind of looked at the different processes that can get them in and out of this cell. Now here is an example of a question from 2002 that's focusing on diffusion. Take a second, pause the video, read the prompt, and then in the upcoming slides, we'll work on answering part A and part B. Now, something to point out from this prompt is that they're giving you all kinds of different data. They're giving you all kinds of different numbers. How we were using 4 different solutions, and each of those solutions was put into a different dialysis tubing. So this was put into a tubing, this was put into a tubing, this one and this one were all separate. And then they were submerged in a beaker containing water. Then they talk about measuring the amount of sodium chloride in the water outside the bag. And then they tell you, but wait, we're only focusing on the results from the 5% bag. So we're ignoring these and we're ignoring this, and we're just focusing on what happened in this particular part of the experiment. So as you look at the data, it might take you a second to go, okay, wait, what am I looking at? So I want you to visualize for me that we have one of those dialysis tubing experiments set up. We have the tube, and in this case, we were talking about 5% sodium chloride, and this bag is submerged in water. Now, before this experiment, there's no sodium chloride out here in the water. What this data shows you is that every 40 seconds, the experimenter is measuring how many milligrams per liter of sodium chloride has entered the water. So thinking about that, where'd it come from? Left, right? So the sodium chloride is leaving the bag. And if we just give a general trend here, we're seeing that as time progresses, we're seeing more and more sodium chloride leaving the bag. So if you're looking at this year's exam, a question like this, they would probably give you this big blob of words. They would give you, instead of having you graph something, they would give you the data, and then they would give you the graph, which I have here. So that's kind of what I would expect if you're thinking about this year's test. So on our axis, we have time, and on this axis, we have how much sodium chloride is measured in milligrams per liter. And you can see that all of our points are plotted over the 160 seconds. So that would be what they would give you on this year's exam. Now, part B, this is probably what they would ask you to write about. So let's go back and look at what part B asked. Using the same set of axes, draw and label. Now, again, they're not going to have you draw anything, but they might ask you to explain. So what would the lines look like if we're now looking at the other three solutions? Okay, so that kind of would be maybe what you would see. Now, lower concentration, so lower numbers, 0%, 1%, and then we'll start there, we're going to see less sodium chloride in the bag, right? As the numbers get higher, as the percentages get higher, we're looking at more sodium chloride in the bag. So that's what this first question is asking you to think about. How would you plan that response? Then we're going to say, based on these numbers, are we going to see more sodium chloride coming out of the bag, or are we going to see less sodium chloride coming out of the bag? So thinking about this, 0% sodium chloride, there's none in there. So what would your graph look like? Look like this, right? There's no sodium chloride in the bag, so we wouldn't see any coming out. 1% solution, so this is our 5%, this is our 0%. Where would you predict 1% to be? Because 1% is greater than 0 but less than 5, we would maybe see something like this. So in your response, you would say the line would be lower than 5% but higher than 1%. And then our 10% bag started with double what our 5% bag had. So we would see an increase of the sodium chloride a lot higher. And again, this is just a quick sketch, not necessarily accurate as far as numbers. So if you look at the rest of this part, this is your rubric that kind of explains what they would look for on the AP exam. So pause the video here, take a look at this, and then in the next video, if you peek ahead, we're going to be reviewing water potential. So have your formula sheet ready, because we'll take a look at those types of problems in our next video.